Hello students, today we will learn about immunity. So, in this video, we'll discuss about innate immunity, type of innate immunity, factor influencing innate immunity, mechanism of innate immunity, acquired immunity, under acquired immunity. We will study about active immunity and passive immunity, and we'll study about miscellaneous immunization like combined immunization, adaptive immunization, local immunity and herd immunity. So let's begin with immunity. First of all, we'll discuss about immunity. What do we mean by immunity? So let's begin with the term. Immunity is defined as resistance exhibited by the host against any foreign antigen, including microorganisms. This resistance plays a major role for prevention of infectious diseases. Immunity may be innate or acquired. Now let's know about innate immunity. It is the resistance which individual possess by birth. It is by virtue of his genetic and constitutional makeup. It does not depend on prior contact with foreign antigen. It may be non-specific when there is resistance to infected infections in general, or specific when it's resistant to a particular pathogen is concerned. Now let's know about type of innate immunity. First is species immunity. It refers to the resistance to a pathogen shown by all members of a particular species. Example: bee anthracis infects human beings but not chickens. The mechanism of such type of immunity is not clearly understood. The physiological and biochemical differences between tissues of different host species may be responsible for species specific resistance. Now, let's know about racial immunity. Within one species, different races may exhibit differences in susceptibility or resistance to infections. This is termed as racial immunity. Algerian sheep is highly resistant to infections. Algerian sheep is highly resistant to anthrax, which is a common disease of other races of sheep. In human beings, it is reported that American Negroes are more susceptible than American Negroes are more susceptible than the white race to tuberculosis. Such differences in humans could be due to socio-economical economic status. However, in a few instances, genetic basis of racial immunity has been proved. Races with sickle cell anemia prevalent in Mediterranean coast are immune to P. falciparum infection, a genetic abnormality of erythrocytes confers immunity to P. falciparum because such red blood cells cannot be parasitized by these parasites. Persons with hereditary deficiency of glucose 6 phosphate dehydrogenase are less susceptible to P. falciparum. Now let's know about individual immunity. Resistance to infection varies with different individuals of same race and species. This is known as individual immunity. The genetic basis of individual immunity is evident from the observation that homozygous twins exhibit similar degrees of susceptible or resistant to tuberculosis. In contrast, such correlation is not seen in heterozygous twins. Now, let's know about factors influencing innate immunity. First is age. The two extremes of life carry higher susceptibility to various infections. In foetus, the immune system is immature, whereas in old age, there is gradual waning of immune responses. The fetus in utero is generally protected from maternal infections through the placental barrier. However, some pathogens cross this barrier and lead to congenital malformations. In some diseases such as chickenpox and poliomyelitis, the clinical illness is more severe in adults than in young children. This may be due to more active immune response which causes greater tissue damage. Now let's know about hormones. Certain hormonal disorders such as diabetes mellitus, adrenal disinfections, dysfunctions and hypothyroidism enhance susceptibility to infections. Staphylococcal sepsis is more common in diabetes, which may be associated with increased level of carbohydrates in tissues. Cortico- Corticosteroids depress the host resistance by its anti-inflammatory, antiphagocytic effects and by inhibiting antibody formation. The elevated steroid level during pregnancy may be related to higher susceptibility of pregnant women to many infections. Now let's know about nutrition. Malnutrition predisposes to bacterial infections. Both humoral and cell-mediated immune responses are reduced in malnutrition. Now let's know about mechanism of innate immunity. First we will begin with epithelial surfaces. First is skin. Under epithelial surface, first sub subheading is skin. It is not only act as a mechanical barrier to the microorganisms but also provides bactericidal secretions. The high concentration of salt in drying sweat, the sebaceous secretions and long chain fatty acids contribute to bactericidal activity. The skin may be fried of bacteria deposited on it, but the bacterial flora normally present on skin is not easily removed even by washing and application of disinfectants. The resident bacterial flora of skin and mucous surface help to prevent colonization by pathogens. Alteration of normal resident flora may lead to invasion by extraneous microbes and thus causing serious diseases. Clostridial enterocolitis following oral antibiotics is one such example. Now let's know about the second subheading, that is respiratory tract. The inhaled particles are arrested in the nasal passages on the moist mucous membrane surfaces. The mucous secretions of respiratory tract act as a trapping mechanism and hair like cilia propels the particles towards the pharynx where it is swallowed or coughed out. The cough reflex acts as an important defense mechanism. Some particles which manage to reach our alveoli are ingested by phagocytes present there. Now let's know about intestinal tract. The mouth purges saliva which has an inhibitory effect on many microorganisms. Some bacteria may be swallowed and are destroyed by acidic pH of gastric juices. The normal bacterial flora of intestines further exert a protective effect by preventing colonization of pathogenic bacteria. Now let's know about conjunctiva. Tears have a major role by influencing away bacteria and other dust particles. In addition, lysozymes present in tears has a bactericidal action. Now let's know about urinary tract. The flushing action of urine eliminates bacteria from the urethra. The acidic pH of vaginal secretions in female due to the fermentation of glycogen by lactobacillus renders vagina free of many pathogens. In males, semen is believed to contain some antibacterial substances. Now, let's know about antibacterial substances. Besides specific antibody formation, there are number of non-specific antibacterial substances present in blood and tissue. These substances are properdin, complement, lysozyme, beta lysine, basic polypeptides like leukins and leukins from leukocytes, plaquins from platelets, and interference which produces antiviral activities. The complement system plays an important role in the destruction of pathogenic microorganisms that invade the blood and tissue. Now, let's know about cellular factors. Once the infective agent has crossed the barrier of epithelial surfaces, the tissue factors come into play for defense. When an infective agent invades tissue, an exudative inflammatory reactions occur by accumulation of phagocytes at the site of infection and deposition of fibrin, which entangles the organisms to act as a barrier to spread of infection. Phagocytic cells ingest these organisms and destroy them. These phagocytes cells are classified in as macrophages, example polymorphonuclear leukocytes or neutrophils, and second is macrophages. One is microphages, second is macrophages, example mononuclear phagocytic cells. Phagocytic action can be divided into four stages. One is chemotaxis, attachment, ingestion, like this. First is chemotaxis. Let's discuss one by one. Chemotaxis: phagocytes reach the site of infection attracted by chemotactic substances. First is attachment, second is attachment. The infective agent gets attached to phagocytic membrane. Third is ingestion. Phagocyte engulfs the infective material into a vacuole. The membrane of phagosome fuses with a lysosome to form a phagolysosome. Now come to intracellular killing. Most bacteria are destroyed in the phagolysosome by hydrolytic enzymes of lysosomes. However, some bacteria like M. tuberculosis, M. leprae resist this type of killing and can multiply within phagolysosome and thus produce disease. Phagocytosis in such instances may actually help to disseminate infection to other parts of the body. A class of lymphocytes called neutral killer, that is NK cells, play an important role in non-specific defense against viral infections and tumor. Now let's know about inflammation. Inflammation occurs as a result of tissue injury or irritation initiated by the entry of pathogens or other irritants. It is an important non-specific defense mechanism. Inflammation leads to vasodilation, increased vascular permeability, and cellular infiltration. Microorganisms are phagocytosed and destroyed. Due to increased vascular permeability, there is an outpouring of plasma which helps to dilute the toxic products present. A fibrin barrier is laid to wall of the site of infection. Now let's know about fever. A rise in temperature following infection is a natural defense mechanism. It destroys the infecting organisms. Fever also stimulates the production of interferon which helps in recovery from viral infections. Now let's know about acute phase proteins. Following infection or injury, there is a sudden increase in plasma concentrations of certain proteins. These include C-reactive protein, that is in short CRP, mannose binding proteins, and many others. CRP and some other acute phase proteins activate the alternative pathway of complement. They are believed to prevent tissue injury and promote repair of inflammatory lesions. Now let's know about acquired immunity. The resistance acquired by an individual during life is known as acquired immunity. It is of two types. One is active and second is passive. Let's about active immunity. Active immunity is subdivided into two types. One is natural and another is artificial. Natural through clinical or subclinical infection. Artificial that is induced by vaccination. It is resistant. It is a resistance developed by an individual as a result of contact with an antigen. This contact may be in the form of natural infection or by vaccination. It leads to stimulate the immune apparatus to form antibodies and or the production of immunologically active cells. Active immunity develops after a latent period which is required for immune system to act, but once developed the active immunity is long lasting. And now let's know about the mechanism. Active immune response stimulates both humoral and cell mediated immunity usually in parallel. Now let's know about humoral immunity. It is antibody mediated immunity. It depends on the synthesis of antibodies by plasma cells. These cells produce specific circulating antibody which combine specifically with the antigens and modify their activity. This modified activity may be in the form of lysis of antigen molecules. Their toxin may be neutralized or in the form of removal of antigen by phagocytosis. Now let's know about cell mediated immunity. It depends on T lymphocytes developed against certain antigens. Antibody synthesis also occurs in response to these antigens, but their role is limited. The cell mediated immunity by sensitized T lymphocytes is important in resistance to chronic bacterial infections. In these chronic infections, organisms can multiply and survive in phagolysosome, that is intracellular parasitism, and in viral infections. Now let's know about the types. Natural active immunity. It is acquired by natural subclinical or clinical infections. Such immunity is long lasting. Persons recovering from smallpox infection develop natural active immunity. Now let's know about artificial active immunity. It is produced by vaccination. The vaccines are prepared from live, attenuated, or killed microorganisms or their antigens or toxoids. In killed vaccines, the organisms are killed by heat, formalin, phenol, and alcohol. These are preserved in phenol, N-merthiolate, and alcohol. Toxoids are prepared from bacterial exotoxins inactivated by formalin or by alarm. Toxoids are immunogenic but not toxigenic. Now let's know about the vaccine. First is live vaccine. BCG for tuberculosis, TY21A for typhoid, Sabin vaccine for poliomyelitis, MMR vaccine for measles, mumps, rubella, 17D vaccine for yellow fever. Second is killed vaccine. Under this, TAB for enteric fever, killed cholera vaccine, Salk vaccine for poliomyelitis, neural and non-neural vaccines for rabies, hepatitis B vaccine. Third type of product is bacterial product, that is tetanus toxoid for tetanus, diphtheria toxoid for diphtheria. Now let's know about passive immunity. Passive immunity is subdivided into two types. One is natural, another is artificial. Natural, that means through transplacental maternal IgG antibodies. Artificial, that means through antiserum injection. Passive immunity is induced in an individual by performed antibodies against infective agent or toxin. This antiserum is prepared by injecting infective agent or toxin in another host. The immune system has no active role in passive immunity. Protection starts immediately after transfer of immune serum. There is no latent period as present in active immunity. Passive immunity is short lasting but is useful when immunity is required immediately. Passive immunity is against of two types, natural and artificial. Now let's know about natural. It is transferred from the mother to fetus or infant. Transfer or maternal antibodies to fetus. Transplanted transplacentally and to infant through milk protects them till their own immune system matures to into function. Now come to artificial. This is through parental administration of antibodies. The agents used for artificial passive immunity are hyperimmune sera of animal or human origin, covalent sera, and pooled human gamma globulin. The oldest method is to employ horse hyperimmune serum. Anti-tetanus serum is prepared by injecting a series of doses of tetanus toxoid to horses and bleeding them to separate the serum. As ATS is a foreign protein, it is liable to cause hypersensitivity reactions. To eliminate these complications, human ATS is employed. This is prepared by hyperimmunization of human volunteers with tetanus toxoid. Protection with human ATS lasts longer as there is no immune elimination of the human globulins. Convalescent sera contain high level of specific antibody and therefore is employed for passive immunization against measles and rubella. Sera of healthy adults can be pooled and is used for passive immunization against common infectious diseases prevalent in the region. Pooled human gamma globulin are used for passive immunization against some viral infections. Example, hepatitis A. It has to be ensured that all preparations from human sera should be free from the risk of infections with hepatitis B, hepatitis C, hepatitis D, HIV and other viruses. Uses of passive immunization. Let's know about uses of passive immunization. To provide immediate short-term protection in a non-immune host faced with the threat of a serious infection. Second is for suppression of active immunity which may be injurious. Example is to use Rh immunoglobulins during delivery to prevent immune response to Rh factor in Rh negative mothers with Rh positive babies for treatment of serious infections. Now let's know about the miscellaneous immunization. First is combined. Under this we will study about combined immunization. Combination of active and passive immunization is employed simultaneously, which is known as combined immunization. Passive immunity provides the protection necessary till the active immunity become effective. Second is adaptive immunity. Injection of immunologically competent lymphocytes is known as adaptive immunity. Instead of whole lymphocytes and an extract of immunologically competent lymphocytes can be used. This is being attempted in therapy of lepromatous leprosy. Such lymphocytes are collectively known as transfer factor. Third is local immunity. Natural infection or the live viral vaccine administered orally or intranasally provides local immunity at the site of entry such as gut mucosa and nasal mucosa respectively. A special class of immunoglobulins plays an important role in local immunity. One type of IgA called secretory IgA is produced locally by plasma cells. Now let's know about fourth number immunity, that is herd immunity. It refers to overall resistance in a community. When herd immunity is low, chance of epidemics increase in on introduction of a suitable pathogen. Eradication of any communicable diseases depends on development of a high level of herd immunity rather than a immunity in individuals. Any questions? Finally finished this chapter. Any questions regarding immunity? You can ask me in the comment box. I'll definitely answer the question and subscribe my channel for more updates. Thank you.